everybody. Let's get started. Good afternoon, folks, and welcome to the Lunchtime Discovery Series. I'm your host, Chris Smith, here at the North Carolina Museum of Natural Sciences. It's good to be with you for another edition of our program today. Of course, we gather here on the museum's YouTube channel every Wednesday at noon for this joint partnership presentation event. This program is brought to you by the North Carolina Office of Environmental Education within the Department of Environmental Quality, and then broadcasted to you and hosted by us here at the Museum of Natural Sciences. It works out really well because our offices are right next to each other, although that's not so big a deal anymore in the days of Zooming and live streaming. But it means that we're able to work together to bring you a great program every single week. And it's gotten even more exciting lately because I'm not at home anymore. I'm actually broadcasting to you from the studio inside the museum. How cool is that? It's really nice to be back. And this place has much better sound quality than the spare room in my house. You might agree on that as well for all the folks who've been tuning in the last several months. But hey, always good to be with you for Lunchtime Discovery. So I hope that you've got your lunch because it's time to do some discovery. Now, as we go throughout today's presentation, I want to remind everybody that you have the option to jump into the chat on YouTube or the comments on Facebook, wherever you're watching, and leave questions, comments, thoughts, insights, experiences of your own as we go throughout today's presentation. After our guest speaker gives their talk, I'll be looking to your questions and comments in order to have discussion. I'll read off the questions for our guest. We'll get the answers. We'll have conversation, discussion, dialogue. It'll be great. The Lunchtime Discovery Series presentations are interactive in that way. So please jump into the chat and interact with us. Now, let me bring on today's guest. I'm excited about today's guest because in the world of science and nature and conservation and education, we tend to talk a lot about big ideas, and we tend to talk a lot about big wildlife, right? Like the cute, fluffy things. But how often are we really talking about the things that are around us all the time, plants, and what's going on with all of the plants that are around us? What are they doing? What's going on in their life? We tend to talk about the things that are just a little bit more like us. So today's guest is going to shed some insight and expertise on the world of plants. Lauren Maynard is an ecologist and a PhD candidate at Virginia Tech and is here to talk to us a little bit about the secret lives of plants. Lauren, welcome to the show.
Hey, great to be here.
So I'm excited to hear a little bit more. Looking at the description for today's talk, kind of some interesting stuff. You're talking about like plants actually doing more than just photosynthesizing.
Of course, yeah, plants do a lot of crazy things and they interact with so many different organisms. So I put in my blurb that maybe caught you today, beetles, bugs, bats, and birds. And so we're going to talk about all of that and how they interact with plants.
Well, I'm excited to learn more and see your presentation. So I'll go ahead and turn it over to you.
Okay. Bear with me all.
Sure, and I'm just looking at the chat on YouTube, and it looks like Marty from the Office of Environmental Education got his lunch in. He's having a North Carolina delicacy, a nab. I'm surprised he's not having some peanuts and a Pepsi.
Okay. Can everyone see that?
Looks perfect.
Perfect.
Looks great. Yep.
Okay, so as Chris said, I'm Lauren Maynard. I'm a PhD candidate at Virginia Tech. I'm in the Department of Biological Sciences, where I study ecology and evolutionary biology. I'm also a graduate fellow for National Science Foundation and for the Virginia Tech Interfaces of Global Change. And today we're going to delve into the secret lives of plants and look at their wonderful world. So the first thing I'm going to start out with, what if I told you that most organisms on land will take flight during some part of their life? So the first thing you think of is what? Things with wings. So that's bats, birds, butterflies, but plants do too. So plants are rooted in the ground, but they've evolved mechanisms of flight for survival and reproduction. And these include both pollination and seed dispersal. These are two examples of wind dispersed pollen and seeds. So on the right, you have milkweed plant dispersing ripe seeds into the wind. So you have the fluffy things at the end, like a dandelion, so they'll take flight to the wind and then go where they may. And on the left, you have a male pine cone sending pollen to the air, hoping it will reach a female pine cone, but we all know that that is usually not where it ends up. A lot of it ends up everywhere else, on our cars, on us, on our houses. And so we know that wind dispersal is distance limited and it's not selective, and it can be messy and it can be wasteful. And so plants have found another way. Instead, some plants have developed relationships with flying organisms, and these include nectar eaters. So the nectar eaters become pollinators, and then the fruit eaters become seed dispersers. So in the top two pictures, we have vertebrate pollinators, including a hummingbird on the left and a nectar eating bat on the right, and all of the yellow in his face is pollen. So he went in to get the nectar. He ended up with a bunch of pollen. He's going to go spread that around to all the other flowers. And then on the bottom, we have two seed dispersers, including a blue-gray tanager eating a bright red fruit, and then a bat on the right who's about to swipe a pepper fruit. So those are two examples of direct mutualisms, or relationships that benefit both parties. So in these scenarios, the animals are providing a service, either pollination or seed dispersal, and then the plants are providing a reward. But there's another side to this, right? Not everything is butterflies and sunshine. We also have antagonists. Antagonists are organisms that want to exploit the plant's resources without offering that service in return. So these could be herbivores who are going to eat the plant, usually the leaves, or pathogenic organisms. And so you see these two sides of the relationship. There's attraction and deterrence, friends and foes. But again, plants are rooted in place. They don't have the means that we resort to, as yelling or running or waving over hay. So they have to do everything rooted where they sit. So how do they do that? They use chemistry. Plants are chemical factories producing an array of compounds to help them interact with the environment. And because these interactions are so diverse, they produce a diverse array of compounds. So let's look at a few. And I'm going to have a few questions here. If you want to answer in the chat, go ahead. Usually this is a little more interactive. So if I show you this picture, can anyone tell me what this plant is? It's common to the Southeast. I've stepped in a lot of it this summer.
Oh my goodness, Lauren. I think everybody's going to know this one in the chat.
Everyone knows this is poison ivy. Yeah. And so what do you start, when you see this plant, what do you immediately start feeling? You feel a little itchy. And this is called by the compound uretiol. As many of us know from personal experience, it has some pretty nasty effects. And the plant uses this as a deterrent. So things don't want to touch it, don't want to eat it, don't want to harm it. Switching to the other side, when I show you this photo of a lily, what comes to mind? Maybe the scent? So floral aromas are the product of numerous volatile compounds working together to attract pollinators. And plants use different floral shapes, colors, and chemistry to attract specific pollinators. So here's a little schematic of roses and violets and lilies and all the different compounds they may use to bring things in. They also will change their shape. And so the floral tubes are formed here on the left, so it has to be really long, and something with a long snout and tongue has to really get in there to get the nectar and also the pollen. And then on the right, you have the petals are forming more of a little hidey hole, so only a small insect can get in there, get covered in pollen while it gets the nectar, and then go again to the next flower. Okay, when I look at these, what do you think of? Hot sauce, maybe some spicy food. So the reason that chili peppers are spicy is due to a compound called capsaicin. And for humans, because we're mammals, we feel the heat. However, birds don't have the receptors for these compounds. They eat the chilies all day and they cannot have that burning sensation. So why do we? So they found that when mammals, and this is usually rodents in its natural system, when they eat the fruits, they destroy the seeds either with their teeth because they're gnawing on it or through their gut passage because it's too caustic. And so by the time the rodents deposit or otherwise relieve themselves of the seeds won't germinate. And so they can't become a new plant, which is the whole point of a seed disperser, right? The whole point, the plant creates this nice fleshy fruit, and so that an animal will eat it, ingest it, and then take it somewhere else because it doesn't want all of its plant babies growing around it, creating competition. So when you have an animal that's taking the fruit, taking the seed, but then not depositing it somewhere else to make a new plant, we call that a seed robber, and that is not what a plant wants. And so birds, on the other hand, can eat the fruit and pass the seed. And so they are really good seed dispersers. And so the plant has evolved capsaicin, that compound, to deter the fruit-eating mammals so they get spicy mouths. And then the compound doesn't deter the effective seed disperser, the birds. But today we're going to talk about a different kind of pepper, piper. So always next to your salt shaker, you'll usually find a pepper shaker, and that's black pepper, or Piper nigrum. The compound that gives black pepper its kick is called piperine, and it's led black pepper to be one of the most important spices in the world. Pepper cultivation began thousands of years ago in Southeast Asia, and once humans took to the seas and began trading, it became an important commodity. They used to call it black gold because it was so important they could actually use it as currency. And today it is the most important spice worldwide, but we usually take it for granted because it's always sitting there next to the salt shaker. The protagonist of our story today is Piper Cinctapholesis. So it's in the same genus as Piper nigrum, but it is a different species. It's a shrub found in the Neotropids. It's a first successional plant species, which means if there's a disturbance, either man-made or natural, it's going to be one of the first plant species to come in and colonize the area. So it makes it really important for restoration. It's also an important food source for both bats and birds. So ecologically, it's important for wildlife. To study this plant, I worked at La Selva Biological Station, which sits along a tropical wet forest in Costa Rica. And since plants are rooted in place, we have to go find them. So that means that a big part of my job as an ecologist is to hike around the forest and find these plants. The overwhelming biodiversity in the tropics magnifies its chemical diversity, which means that studying chemical ecology can be tricky because most secondary metabolites are undescribed and have numerous potential benefits. In our paper we published last year in Ecology, Maynard et al., 2020, we embarked on this process of discovery by tackling 4 questions. And that's what, where, when, and how. What compounds are in the plant? Where are they occurring? When are they occurring? And how are they serving the plant? So to give you a little backdrop on our protagonist, Piper Cinctapholesis, the fruits ripen distally along the branch, and so each spike is either an inflorescence or an infructescence. And an inflorescence is hundreds of little flowers, and once they're pollinated by flies, they turn into single druplets, and then the spike is an infructescence. But those are both mouthfuls, so I'm going to say the words flower and fruit when I'm talking about those structures today. Bats in the genus Curator are what we call Piper specialists. They rely on these fruits for their main source of food throughout the year, and their nightly diet consists of 40 to 100% Piper fruits. And so here on the right, we see a Curator bat about to swipe a ripe Piper fruit. Many bird species eat Piper fruits, but there are no specialists, and we call these generalist feeders. So they eat a variety of fruits, insects, and seeds. And here on the left, we see a male Passerini tanager eating a Piper fruit. You can also tell the difference by how they eat the fruit. You see the bird perched and eating it next to the plant, whereas the bat swipes the whole fruit. And to give you an idea of the consistency when they're ripe, they're very smushy, kind of like kiwis. Bats really dig them, so even in the hand, they're going to go at it. And when they're not in the hand, they eat it like a corn on the cob. I hope this is streaming well. I know sometimes these videos come through glitchy, but he's holding it with his thumbs, which what we see as the claws at the top, and he'll turn it around like a corn on the cob. He'll go all the way down the fruit, skimming it. Once he gets to the end, he'll go back around and make sure he didn't miss any bits. He's about to turn back around now. So Piper Cinctapholesis has its own suite of antagonists and mutualists. In addition to the bat and bird seed dispersers that I just told you about, they also have pathogenic fungi that will attack the fruits. So who will get to the fruit first? Is it going to be the mutualist seed disperser or the antagonistic pathogenic fungi? And how is the plant going to help mediate that race between the two? Let's go back to our 4 questions. What, where, when, and how. To start to answer this, we have to do compound extraction and analysis. So as I said, the first thing we do is go out to the field and collect these plants. After that, we dry them and ground them into a fine powder. So we see the first, that's the first one on the left. Then we add solvent for extraction. That's our second step. So we add the solvent, and just like any time we brew coffee or steep tea, the solvent or the liquid is going to pull the compounds out of that plant material and into the liquid. The next step is filter. So just like you don't want a cup of coffee with grounds or a cup of tea with tea leaves, you want just the liquid. So after the solvent has extracted all those compounds from the ground tissue, we're going to filter all of that out. So we just have the pure compound in the liquid. And then we put that on an instrument for analysis. And for the things that we did for this project, we used a couple of different things, including NMR, or nuclear magnetic resonance, or GCMS, which is gas chromatography mass spectrometry. So what are the compounds? This work was done by our collaborators at the University of Nevada, Reno at the Hitchcock Center for Chemical Ecology. So using NMR, they identified 10 dominant compounds in the class alkenophenol, which are shown here. And as you can see, the compounds consist of alkenyl chains bound to a phenol. And the differences in the compounds come from the rotations of the bonds in those chains. For our second question, where do they occur? Here we have a graph with the y-axis as our total alkenophenols, and then on the x-axis, 5 different tissue types, including vegetative and reproductive. And so we found that the unripe pulp had the highest concentration of the compounds. This was followed by ripe pulp, then flowers, and then at barely detectable levels, we found it in mature leaves and seeds. Out of the 10 compounds, we found that the flowers and the pulp had all 10. However, only 2 of the 10 compounds were found in the leaves and in the seeds. So fruit pulp had higher alkenophenol concentrations and diversity. For our next question, when do they occur? Again, on the y-axis, we have our total alkenophenols, and on the x-axis, we have our developing tissue. So this is what it would look like biologically. This is going from our developing flower all the way through to our ripe fruit at the end. And so here, each colored line is an individual plant as it ripens. When we were fitting these data, we found that not the linear, but the nonlinear fit was a better fit of these data. So the concentration of alkenophenols were being allocated non-linearly across development. They weren't going straight up, but they were actually increasing and then leveling off to a decrease at the end. For our last question, how do compounds serve the plant? We'll bring back our antagonistic fungi and mutualistic seed dispersers. To test these interactions with antagonists, we performed a microdilution assay. And these assays were done by our collaborators at the University of Guelph and the Organization for Tropical Studies. 3 of the most common fungal taxa found on the seeds were selected, and these included 2 Fusarium species as well as Microdocium lycopodium. So on the x-axis, we have alkenophenol concentration, and on the y, we have the average absorbance, which is a proxy for fungal growth. And so what did we see here? You see 2 lines of Fusarium and the lycopodium are going down, as well as, but Fusarium B doesn't seem to be affected. So alkenophenol showed a clear antifungal activity against 2 of the 3 fungal taxa. So they're functioning to deter these fungi from attacking the fruit. Lastly, we observed and experimented with the vertebrate seed dispersers of Piper. First things first, we got to observe it in the field, see what it's doing, make sure it's actually interacting with the plant. So that's an ecologically relevant question. We measure the number of fruits removed during the day and during the night, and whether the fruits were ripe or unripe. We saw greater nocturnal removal and less diurnal fruit removal with a larger proportion of unripe fruits. So what does that mean? Bats removed about 11 times more fruits than birds, and birds removed a higher proportion of unripe fruits. So birds didn't remove many, but the ones that they did were actually the unripe ones and not the ripe fruits. Then we did a flight cage study. So the first things first, we have to catch bats and birds. Does anybody know how to do that? Just like run at them really fast and catch them with your hands.
Or just hold on to some Piper fruits and see if they come come down for you?
Yeah, exactly. Ambush them, get a blind. No, we use mist nets. So we set up these mist nets and see if they're going to come and hang out with us. So unlike plants, we actually have to set up something and catch them and wait for them to come for us. So these mist nets are in between two poles, and we hang up the nets. They're a very thin mesh. And as you can see on the picture on the right here, they have these trammels that they'll fall into. So hopefully they don't see the net, they'll fly into it. It's very soft, and then they kind of get swaddled in that little, what we call a trammel, or that little pocket. And then we wait. And then once we see that they're there, he's hanging, this is a sparrow hanging out in his trammel. A trained professional will come out and extract him. And here's a Carolina chickadee in the hand. Both these pictures were taken at the Prairie Ridge Eco Station out in Raleigh. And the same thing happens with bats. So you're going to see a bat hit the net here. And then you're going to see my friend Paola come and extract the bat. So it's the same process. You just do it at different times of the day, different trainings, and also a handful of different precautions you have to take. Don't do this all. Okay, so we put everything in a flight cage, not at the same time, one bat and one bird at a time. And we had a treatment dish, which was food supplemented with these compounds, and then a control dish, which had food that did not have any of the compounds. And so bats usually made a pretty obvious choice. They would fly around trying to decide which one they wanted. And then once they decided, they would just kind of faceplant most of the time, pick it up and leave. So what do we find? So here on the y-axis, we have the amount eaten, and on the x, we have control, and then that sign here is the alkenophenol. So it's the unsupplemented or the supplemented diet. On average, bats avoided alkenophenols in favor for the food that didn't have any compounds, whereas birds really didn't seem to mind. So alkenophenols deterred bats, but did not deter birds. So let's put all that together. We answered our 4 questions. What's there? We found 10 alkenophenols in Piper Cinctapholesis. The highest concentration and diversity was found in the fruit pulp. We found that the alkenophenols had a nonlinear allocation across ripening, where it increased and then decreased as the fruit became ripe. And then how was it serving the plant? It was antifungal, so it stopped the growth of fruit fungi, but it also made it less tasty to bats. So our results are consistent with the defense, it's allocated by defense allocated, sorry, the defense is allocated by fitness. And so we see here on the bottom again, development and phytochemical investment, where it increases until the seeds are mature and it's ready to leave the plant. And then all those defensive compounds are going to stop dropping off. So they're in the fruits, they protect the fruits. Bats don't like it. And so that's why we think it's a possible explanation for that decrease during ripening. It's a selective pressure exerted by bats, which we believe are probably the more the better seed disperser. So like we saw with the chilies, where the mammals were deterred, but the birds were not, we think this is flipped with this. So now the bats, the mammals, are the better dispersers and the birds are not. And so that's what's exerting this downward trend. Okay, that was a lot of information. So let's take a deep breath, stretch break. We're going to refocus our attention on the broader picture of chemical ecology, because some of you, hopefully not all of you, but some of you might be thinking, okay, I just learned a lot of information about a very specific group of compounds and a plant I've never heard of, but bats I'll never see. Who the heck cares? Hopefully not all of you think that. But the truth is, there's still so much we don't know about phytochemical diversity around us. And this study was designed to be also a roadmap for future scientific endeavors. So not every scientific discovery will have an immediate impact on human society. But to even get to that point, we need to know what's there and how it evolved to function in its natural environment. So from the study, we know that these alkenophenols are effective deterrents of some fusarium fungi, which commonly affect food crops. So this could be a new agricultural treatment to reduce crop loss, which is really important, especially now. And we would never have harnessed the power of hot sauce or black pepper without first answering these 4 questions. So aren't your taste buds glad that we did? So we're going to shift here. I showed you the picture of the bat sampling the food and figure out which one you want. So how do bats know which fruit is ripe and which flower is open? Bats are famous for one of these, but there might be one you might not thinking of. Echolocation and olfaction. So bats are famous for being one of the only animals to use echolocation. They do this by sending out ultrasonic ticks, and then they determine their surroundings based on the returning sound waves. They also use olfaction, and olfaction is the sense of smell. So this is a bat metaphorically smelling roses. So we know that fruit and nectar eating bats use both echolocation and olfaction to find open flowers and ripe fruits. And so usually you have a bat echolocating, he's gonna maneuver through his surroundings, and then he's gonna find, we'll say this is a Corollia, finding a piper tree. It finds the tree using echolocation, and then it starts sniffing around it, and it uses its nose to find the ripe fruit. And so the ripe fruit changes its volatile profile. It also gets softer, as you saw in that video where it was kind of like a squishy kiwi. And so it's echolocating, it's sending back that it's softer, it's smelling, it's telling, the plant is telling it that it's ready. And so that's how it knows when it's going to swipe. Flowers do a very similar thing about when their pollen or when their nectar is ready and when they're open, because bat pollinated flowers tend to only open at night. And these are examples of direct mutualism. So pollination and seed dispersal both provide direct benefits to plants. We know that plants directly communicate with the bats to let them know which ones are ready to be visited. Bats also provide indirect benefits to the plant by eating their insect herbivores. So you see there's a negative interaction with the insect eating the plant, and then a positive indirect interaction with the bat coming to eat that insect. So could plants also be indirectly communicating with insectivorous bats? This is what the last part of my PhD is hoping to answer. So plants emit volatiles all the time. However, when they're damaged, they emit something that's called herbivore-induced plant volatiles, or HIPVs. Once a plant is damaged, it changes that scent profile. And you can kind of think of when you're cutting grass and you have the fresh cut grass smell, that's mechanical damage, but you did so much of it, the plant is ramping up its volatiles. So you can really smell that when someone's cutting their grass or cutting flowers or anything like that. And so they change their volatiles also based on how they're damaged. So cutting would be mechanical. This is specific to herbivore induced. So you have an herbivore chomping on this plant. And so why would they change their volatile profile to be different if an herbivore is on the plant? One hypothesis is for the release of HIPVs is that it's a distress call. So we found that insect predators, like these on the right, a ladybug, a parasitoid wasp, even birds, can cue in on the shift in volatiles. And by being called to the plant and removing the insect, we see another mutualism. The animal provides a service by removing that herbivore, and the plant has alerted it there. So we see that indirect communication between the plant and the insectivorous mutualist. But for bats, we're not sure if this is happening yet. We're actually not really even sure how much insectivorous bats use their nose, even though we're very sure that fruit and nectar eating bats are definitely using both of them in tandem, echolocation and olfaction. So that's what I'm hoping to answer. And so the first thing to do to answer this question is to choose the system. So I chose soybeans. Soybeans are pretty important. You might have heard of them. They're important worldwide, globally as a crop. They're also important for my question because they continue to produce volatiles at night. So if you have a plant where its volatile production is linked to photosynthesis and it shuts down at night, then that's not going to be a plant that a bat is interacting with, as bats are only out at night. And to answer these questions, I'm working with the Smithsonian Environmental Research Center and the University of Maryland. So first, I did an experiment with soybean plants where I damaged plants on one side of a field and not on the other side. So we had one field, one with a bunch of damaged plants and one with undamaged plants. And to damage these plants, I used the insect herbivore, the Japanese beetle, pictured here. He's a very handsome invader, and he was very happy to eat my soybeans when my lab-grown caterpillars thought it was just too hot to eat. So he was very helpful this summer. Next, for my another round of experiments, and so instead of the naturally produced HIPVs, I chose to use a synthetic compound, indole. This is in the bouquet that soybeans produce when they're damaged by an herbivore, and we know that this compound specifically is also important in attracting invertebrate predators like wasps. So I assemble these dispensers. It's just a small vial. You can kind of see it here at the top. It's a small vial. I filled with the compound, put the screw on, and on the top I pierce with a microcapillary tube. So it allows for a gradual release of this compound over time. So how did I measure bat response? And what is that green box? So I'll answer both questions with one slide. These boxes are ultrasonic detectors and recorders. So they're triggered by ultrasonic sounds. This is usually bats, but sometimes insects and sometimes flying squirrels. But then I use a program to go through and auto ID the bats based on their calls. So just like birds, bats have distinctive sounds and they produce this sonogram. And so I load it into this program. I look at the sonogram and we can determine, okay, this is a big brown bat. And so that's what I'm going to talk about today, because our big brown bats are the most common bat here in the area. They're also responsible for most of the calls. And also the total activity at this point might not account for everything interacting on the ground. And so a lot of the bats, like including the free-tailed bats, are flying really, really high. And so accounting for their activity, it's going to be in my dome of what I'm counting, but they're not going to be on the ground interacting with plants. So here's a lovely big brown bat, and here's how he interacted with my experiments. So on the bottom, we have damaged plants and undamaged plants, and on the y-axis, we have relative activity. So you see there was a little bit more activity in the damaged plant plots, but the standard error is pretty high on both of these. So that's not a clear effect. It's not statistically significant from my preliminary results. We did have a lot of confounding factors in the field. I mean, if you think about a field in the middle of, nature, just because I damage some of the plants doesn't mean, and don't damage some doesn't mean the other herbivores aren't going to come in. So we had natural herbivory occurring, a lot of deer herbivory occurring. So it was a bit messy. So this signal could be here. This could be a real trend that was swamped out, or it could not be there. And that was just a relic. So we're not sure about this, but the indole trials seem to be a bit clearer. We're seeing a strong signal. So there's clearly more activity in the plots where the indole is being released compared to the plots where we had an empty dispenser. And so from this, we can tell that bats may have at least noticed that the indole was there and are attracted to it. So they're not deterred, they're probably attracted. We don't have any, I'll move to the next one. Are insectivorous bats using HIPVs? That's the question we would answer. So from the data, y'all are scientists now, from the data that I presented to you, what do you think?
I'm gonna say, Lauren, that I'm not so sure.
Yeah,
I'm like maybe a little bit.
Maybe is my answer right now. So still to be determined. I hope to get a more clear answer by the end of the year. So I'm still here in Edgewater, Maryland at the Smithsonian Environmental Research Center collecting these data. The indole data I just pulled off the detectors like 2 days ago. So that's the first time anyone's ever seen those data. And it's exciting, especially since the plant trials weren't showing us anything that was really clear or definitive. So I hope to keep moving that way. And then we're going to start adding in more compounds, right? So that's just one compound. Can we add in different ones? Can we try to add in a bouquet? Are they deterred by some but attracted to others? Just because a plant is emitting it in like copious volumes, it could be serving a different function. And it could be maybe the little compounds that the plant's emitting that are actually really important for this specific interaction with bats. So, and you'll notice I did not use a flight cage study with this experiment, which would be the next step, which would be great. And our global climate working with bats in that interactive way was unsure in the beginning of the year, but hopefully we're moving past that and we can start doing this more interactive work, right? Because that's how you find out those fine scale interactions of, how much do they like this or not like this? So if it's happening, big if, very big if, but what are the possible management implications if bats are using these herbivore-induced plant volatiles? well, this is going back to agriculture. So we can continue to use these as, instead of using these tough herbicides and pesticides, we can actually just go in and maybe attract bats and start attracting things that are going to kill the insects for us without killing the insects. And so this would be an example of an integrated pest management practice that would focus on the attractant versus the killing. And so this will create hopefully an option for a healthier way for us and for the environment to grow food. So we're going to circle back around, sink back into this picture again, deep breaths. We need holistic and integrative studies to really look at these multi-trophic interactions that include both antagonists and mutualists. And in the bigger scope of things, it's important to explore chemical ecology and diversity and the pressures that drive it. So now more than ever, it's really important to conserve that biodiversity. And by doing that, we're conserving the chemical diversity. Because only after we've discovered the compounds and their ecological role can we start to explore their uses in society. So I have a pop quiz. Hope everyone's ready. Do you have to go to a tropical rainforest to study plants? Of course not.
I sure hope not.
No, of course not. You can go to your backyard, especially North Carolina or anywhere in the Southeast, which is what I'm more familiar with. You can go to your backyard. You can go to a local trail, the North Carolina Botanical Garden, the Capital Area Greenway. You can go anywhere. And so you don't have to go all the way to a tropical forest. And we make it seem like science, you have to travel or you have to have some high-tech instruments and it can be intimidating, but it doesn't have to be. And you can start with really simple questions and build a story on that. So I'd like to thank my co-authors for the ecology paper, as well as my Maryland collaborators, John, Ken, and Karen, my advisor, Susan Whitehead, as well as my entire lab group, the Whitehead group, and all the people funding this. And so if you have any questions, there's also my email address and my name. Thanks for tuning in, guys.
Lauren, thank you so much. All right, everybody, let's give Lauren a great big round of applause wherever you're at. Drop your appreciation and clapping hands emojis into the chat for Lauren. Excellent job. Right now, what's populating in the chat is everybody saying, no, you don't have to go to the tropics to study plants. And I think we've even got some classrooms who are tuning in. And so like a whole classroom just yelled out, no, study plants in your backyard. That's great. I love that. And that's a great message for students too, like get out into your schoolyards or into your local parks and do plant studies.
Yeah, a lot of the things that, you've noticed something and you're like, oh, I wonder if anyone's looked at that. And sometimes people haven't, it's almost too close to your face, too close to your nose to know it. So if you see something interesting and you have a question, test the hypothesis. If you have a bird feeder that's red and one that's yellow and you notice one bird's going to one versus the other, then take some data. I don't know, see if that's what's working.
That's how you do the science. Although I would say, though, like in your case, if somebody gives you the opportunity to go to Costa Rica in order to study bats and plants, you should probably take that opportunity.
Yeah, you don't turn that down.
Which looks like it would have been a lot of fun, maybe a lot of work too. I don't know what it's like doing field work out in a tropical forest like that.
Everywhere is different. La Selva Biological Station is really wonderful, and they're set up to do all of these experiments. And so that's where a lot of the tropical ecology research from the 70s and 60s, when they first started doing it, came out. And so they have year-round housing and a cafeteria. And honestly, it is like the Ritz of studying in the field. I've been in other situations where you're living in a tent and, out of a truck and you have to bring all your water and food in. And it's different every time and it depends on what you want to do, but you can definitely find experiences both here. I mean, North Carolina and the Southeast is great for that. And also if you want to go abroad into Latin America or beyond.
Excellent stuff. Folks tuning in, watching right now, I'll remind you, drop your questions into the chat and we're going to get to those here in just a moment because I see some questions that are starting to populate about plants and their interactions. But Lauren, I'm kind of curious, did you find anything in in studying bats and the plants and the chemicals that they're using that was particularly surprising, given that these things have been studied in like in pests, and I think you said it in birds also? Were the bat interactions particularly surprising to you?
Well, for the first part of it, it was surprising because we do think bats are the better dispersers of these piper fruits. And so it was weird that they were deterring the better disperser, because you think that would be kind of backwards, but birds tend to eat almost anything, if you haven't noticed. They're pretty hardy. And bats are, because they are that specialist, they're specializing on one thing. And that's the reason we found too, though, that we have that decrease of the alkenophonol. So, you want to have a miracle compound that does everything. It attracts what you want, deters what you want, but realistically, that's not what's happening. So that's why plants have to have so many. They have to have ones that do the attracting and the deterring, and then kind of battle that out. And so it's over these like millennia of evolution that they've really fine-tuned it. With the bats here, it was surprising that people really haven't looked at their olfaction, at least in North America, or I guess above Mexico, because we only have insectivorous bats here in the Southeast US. So we haven't really, are they smelling? We know that they can smell their roost mates and they're young, but they're not really using that when they forage. Can they smell insects or plants or anything else? So hopefully we can figure that out soon.
Yeah, interesting follow-up questions there, because you brought up bats and olfaction, and I was thinking, I was trying to think of bats that would use their sense of smell, and right, I'm in the Southeast. I was only really thinking about insect-eating bats and not considering that they could use their nose, and of course I was ignoring like fruit-eating bats because they're not in my backyard.
It's easy to forget about them when they're not, in your face.
All right, let me see what questions have come in during the talk, and we'll start throwing those your way. Are you ready?
I'm ready.
Okay, Matt Bertone. Hey, Matt, thanks for tuning in today. Actually has a question about poison ivy.
Oh.
We'll see if you can get this one. How do things avoid poison ivy when the reaction is delayed and there's no warning, especially from roots or vines? What's the evolutionary advantage?
So poison ivy is actually tricky. It's great for talks because everyone knows it like immediately when they see it. But so one of my committee members, John Delusco, is looking at this and how they've evolved uratiol and where they put it, because it's actually everywhere in the plant, which is pretty uncommon with plants. They put it in the leaves and the fruits, and it's in the plant family Anacardiaceae, so it's already just in that crazy family. So he's trying to figure that out, and who it evolved to deter, because you do see a lot of things rubbing against it and eating it, and we don't know.
Maybe I'm thinking about all the different mammals that eat it anyway, and are undeterred by it in the first place.
Yeah, so the mammals eating it, and then the birds eating they have a single-seeded fruit, they have a drooped fruit, and so the birds will eat the fruit. So yeah, it deters us. In nature, we're still not super sure why it evolved and what it's actually doing.
Maybe it's just there for us, Matt. Okay, Glenn is asking about piper fruits. Do the ripe piper fruits have a high sugar content? And is that what makes the bats like to eat them?
They do have a higher sugar content. There's a shift as soon as they become ripe. They get really soft and smushy. And so bats can pick that up on their echolocation because it doesn't jump back as sharply, because it goes into the fruit. It does have a higher sugar content, and I tried to quantify that. I didn't do that in this paper, but it changes texture as well. And actually, you saw the birds ate the unripe fruits and the bats ate mostly the ripe fruits. And there's actually a really big competition for these fruits, especially in the dry season when other fruits aren't available. And they're taken almost as soon as they're ripe, they're taken that night. And if they aren't taken for whatever reason, usually because they're near a building or somewhere where animals don't want to go, it'll start to rot immediately. So that competition, bats, birds, and fungi, is actually pretty tight.
Oh, wow. Okay. Yeah, you had mentioned that earlier, and I was kind of curious about who tends to win. Do you end up seeing more fruits with that have been taken over by pathogens because the pathogens can just more easily move around fruit to fruit, or do the bats
There's
and birds
that
really get
small
lots of them?
window when they're, yeah, there's that small window. So the bats almost always win the competition, but if for whatever reason they don't, the fungi get it pretty quickly.
All right. Okay, Marianne wants to know, are there any particular plants that could be used in a household garden to ward off herbivores, but not pollinators? And Marianne says that their tomatoes survive, but the peppers keep getting eaten before they can get to them.
Yeah, I get asked this question a lot, and the things that I read, people try and they don't, because I think it's like a lamb's ear and marigold, and some of the, it's like, the lamb's ear is fuzzy, or like marigold has some compounds that they don't like to eat, but everything still gets eaten. So I would suggest probably putting in something that is really tasty, so like a honey pot. So you're gonna attract every herbivore to that one plant that you don't care about, and then all your other plants will be safe. So just they're probably going to be there. So just put them on something that you don't mind them eating would be my recommendation.
There you go. All right, let's see, next one. Okay, Kate was asking for some clarification. Right, so are you saying that plants intentionally attract insects that will harm or eat them in order to attract a bird or a bat that will pollinate them?
Yeah, so it's not intentional, but right, they're putting out this advertisement basically that's like, look, I have the resource. So for pollinators would be the nectar. So they're like, yeah, I have this resource, come check me out. And so then they have to do other means to try to, that's the dichotomy, right? So they have to attract certain things but deter others. And so they have tried to many of that evolution to hone in those different compounds, but it's an arms race, just like the fungi and the bats and the birds. So which is going to get it first?
Which is going to get them first. Do you think industrial agriculture's current use of insecticides and pesticides could be interfering with the natural HIPVs that plants would normally release?
It's a good question. When they apply these things, it's usually like they're not trying to harm the plant that it's on, but there's always some byproduct. And just the way that we've domesticated plants too, we've also changed the volatile profile. So, the natural tomato, or what it started as and what the tomato is now, look very different. Their chemistry is very different, and so is their volatile chemistry. And so we may have done a lot of things to be interacting with, or to interrupt that. And yeah, that's a long-winded of saying, yes, we've done a lot of things to kind of put our hand into the evolution of plant traits and our domestication, but we'll see. I'll let you know.
Yeah, that's the next study.
Yeah.
All right. How would plants evolve to put a coating on their seeds if they have no awareness of what happens to the seeds?
So that's through the evolution of it. So if you continue, so there's a few different hypotheses for how plants create these compounds, but one is the evolutionary arms race. And so if they created a compound, so capsaicin, and then most of their seeds that had capsaicin survived because they weren't getting predated by the rodents, then those seeds would continue to survive. And so you have that branching. So then we just get capsaicin that continues to be in that one plant species. Plants are smarter than you think.
I mean, that's the fascinating thing to me about evolution and natural selection, whether you're talking about plants or animals, pathogens, is that it's always these random tangents that species go on and that end up giving us things like capsaicin, or bats that use echolocation that they can use to find ripe fruits the minute they become ripe.
Yeah.
That's astounding. I said nature's wild.
Nature's wild.
Nature's wild. Okay, let's see here. The folks at the Global Change Center at Virginia Tech want to know, how can we help our bat friends?
Okay, that's a good question. So if you're not aware, the bats in the Southeast are suffering from white nose syndrome. So they're already getting hit pretty hard. They were coming back. We were doing better with our pesticides. So they're coming back and now they're getting hit again. And so something we can do, put some bat boxes around your house. They have to stay somewhere. This will keep them out of your house at least. You can also use less harsh pesticides so that if they do eat the insects that ate the pesticide, you're not going to get that biomagnification. We only have the insect eating bats here, so pollinator gardens and things like that don't really help them. But those two things I think could make a difference here, and just supporting things that come across your desk or your ballot, things that are going to protect the wildlife and allocate money to them as well.
I just recently put up a bat house, and I'm eagerly awaiting for the first bats to use it as a roost. I think it might be late in season for it at this point. I didn't get any bats this summer. Maybe next year.
Maybe next year. Yeah.
Maybe next year. I'm keeping an eye on it. All right. Well, Lauren, again, thanks so much for being on the Lunchtime Discovery series today.
Yeah, thanks for having me.
And I just vanished.
I did. Oh, there you are.
We'll use that one. Sorry, everybody. Lauren, thanks so much. Folks, thanks for tuning in to this edition. We will be back here, of course, next Wednesday at noon with another special presentation. Matt Willey from the Good of the Hive Science and Art Project. If you've visited the museum and you've seen the bee mural on the backside of the Nature Research Center, that is his piece of work. He's going to be on the show next week because next week is also Bug Fest here at the Museum of Natural Sciences. So all next week, Monday through Thursday, we're going to be doing special virtual presentations about all kinds of arthropods. Many of the presentations are going to be about pollinators. So we're going to be talking a lot about bees in particular next week. Don't miss out. Lots of great shows. Bugfest.org is where you want to go to see the schedule of events. Register for free for all of the programs that sound cool and interesting to you. It's kind of like a whole virtual conference, but all about bugs and arthropods. So you can just sign up for all the sessions that you want to attend. It's all free. But also while you're there, check out, we've got a couple of cool Saturday celebrations happening on the 18th out at Prairie Ridge Eco Station, in fact, which even featured in Lauren's presentation today. How cool is that? So make sure that you check out bugfest.org and of course follow the Office of Environmental Education. They've got a lot of cool stuff happening all the time, like this program. Their website is eenorthcarolina.org. So from the Environmental Education Office, from us here at the Museum of Natural Sciences, take care, everybody. Stay safe, keep your community safe, and hey, we'll see you again for Bug Fest next week. Bye, everybody.